When someone consumes cannabis, whether by smoking or ingesting it through edibles, tinctures, or other means, the effects are rapid. Cannabis quickly enters the bloodstream and reaches the brain within 30 seconds, permeating throughout the brain and body. Peak concentrations of cannabis are typically reached within 30 to 60 minutes, triggering peak biological effects. These effects usually last between 3 to 4 hours, although individual metabolism, familiarity with the compound, and frequency of use can influence the duration. Factors that can impact the duration and intensity of cannabis effects include individual metabolism, psychological and biological familiarity with cannabis, frequency of use, first time, occasional, or regular, method of consumption. Cannabis components, including THC and CBD, are highly lipophilic, meaning they easily bind to and pass through fatty tissues. Since every cell in the body, particularly neurons, has a double layer of fatty tissue in its membrane, these components can effortlessly penetrate cells. Once ingested, cannabis components rapidly enter the bloodstream, allowing them to immediately access neurons and other cells. They then bind to endogenous cannabinoid receptors, influencing neural signaling and producing the subjective effects of cannabis. Notably, these components can remain in cells for up to 80 days after ingestion, highlighting the potential for prolonged effects on neural function and behavior. Cannabis strains can be broadly categorized into sativa and indica varieties, each producing distinct subjective effects. Sativa strains typically induce mood elevation, increased energy, a head-high sensation. In contrast, indica strains often produce sedative effects, relaxation. The differing effects of sativa and indica strains can be attributed to their interaction with CB1 receptors, which are widespread throughout the brain and body. Sativa strains, in particular, can stimulate neurons, leading to mood elevation, increased creativity, improved focus. These effects are generally dose-dependent, and sativa strains can have varying impacts on individuals. Cannabis's impact on cognitive function is a lesser-discussed topic. The activation of CB1 receptors in the prefrontal cortex plays a crucial role in this process. The prefrontal cortex regulates limbic circuitry, which evaluates internal states, e.g., heart rate, and external stimuli. These systems typically function as threat detection mechanisms. Cannabis, particularly sativa varieties, can create a balancing effect. In the prefrontal cortex, cannabis increases mood, alertness, and focus. In the amygdala, cannabis decreases stress and threat detection. This balancing act can help individuals enter focused states, sometimes referred to as flow states. However, it's essential to note that cannabis does not consistently induce flow states, and its effects can vary greatly. The sativa varieties of cannabis can profoundly impact brain function, shifting users toward a state of narrowed focus, elevated mood, heightened sense of importance, relaxation, increased energy. These effects can be beneficial for creative pursuits, but not necessarily for productivity. However, individual responses to sativa varieties can vary greatly. Some people may experience intense anxiety and paranoia, regardless of dosage, type of sativa variety, that is the type 1, 2, or 3, THC to CBD ratio. Predicting individual responses to sativa varieties is challenging. Despite our understanding of how THC and CBD interact with the brain and body, there is no reliable way to forecast who will experience intense anxiety and paranoia, intense relaxation, focus, and creativity. Research suggests that THC and CBD tend to amplify the effects of various brain systems, rather than producing consistent outcomes across individuals. Instead, individual responses to specific strains or consumption methods tend to be consistent. For example, if someone experiences paranoia or anxiety from a particular strain of marijuana or edible, they are likely to experience similar effects every time they consume that same strain or variety. The cannabis industry has grown exponentially, driven in part by the desire for customized products. Many individuals invest time in finding specific strains and hybrids that work best for them in various contexts. Unfortunately, predicting individual responses to cannabis is complex. There is no definitive way to determine which strain or variety will suit someone based on physical characteristics, personality traits, or other factors. The only way to truly determine how you'll respond to a particular strain is through personal experience. However, it's essential to approach this with caution and responsibility. What we do know is that certain categories of effects, such as increased focus, reduced anxiety, are primarily attributed to the activation of areas like the prefrontal cortex. When individuals ingest or smoke indica cannabis, they often report improved sleep transition times. However, we'll delve into the actual effects of indica on sleep later. Indica cannabis tends to 1. Suppress amygdala activation and threat detection centers in the brain by binding to CB1 receptors and utilizing retrograde signaling mechanisms. 2. Shut down the hippocampus, a brain region associated with memory formation. As a result, indica varieties can cause profound defects in short-term memory, potential long-term memory impairments with prolonged consumption. We'll explore the effects of short-term, medium-term, and long-term cannabis consumption, as well as occasional use, in more detail later. Key takeaways. 1. Mechanism of action. Cannabis impacts the brain and body primarily through CB1 receptors. 2. Variable effects. CB1 receptor activation can accelerate or slow down various biological mechanisms, leading to distinct effects depending on the strain, that is the sativa, indica, or hybrid. 3. Unpredictable individual responses. Despite extensive research, it's impossible to predict how a specific strain will affect an individual. Personality traits, anxiety levels, or other factors do not reliably indicate how someone will respond to a particular strain. Remember, individual responses to cannabis strains are highly variable and unpredictable. Dosing THC and CBD can be more precise with edibles from controlled sources, as they often specify the exact amount of THC and CBD in milligrams. However, this is not always the case with smokable cannabis products, particularly those obtained from unregulated sources that may lack clear labeling. Although the commercialization of THC and CBD products has improved labeling and dosing accuracy in some regions, including the United States, many individuals still consume cannabis from unverified sources. As a result, predicting the effects of cannabis use is challenging. Individuals may experience benefits such as anxiety relief, improved mood and focus, enhanced well-being, pain relief. Conversely, they may encounter adverse reactions like full-blown panic attacks, increased anxiety. Dosing information alone is insufficient to predict these outcomes. While we can establish general categories for cannabis use, such as low, moderate, and high doses, as well as frequent and infrequent use, pinpointing exact safe consumption levels remains challenging. Unlike alcohol and nicotine, where specific guidelines exist, cannabis's effects are highly individualized and context-dependent. Many people wonder about the safe frequency for consuming cannabis, whether through smoking, edibles, or other products. However, defining safe is complex, and there's a lack of standardized controls over the cannabis products available. Even in controlled environments, predicting the effects of THC and CBD is difficult due to individual variability in response. Before discussing the specifics of cannabis use, I'd like to briefly outline the brain areas impacted by THC and CBD. Here's a concise list. When cannabis is consumed, regardless of the THC/CBD ratio, deficits in memory are common. This is due to reduced electrical activity in the hippocampus, a brain region crucial for memory formation, particularly short-term memory. This effect is consistent across sativa, indica, and hybrid strains. Cannabis strains have distinct effects on brain activity. Sativa varieties tend to activate the prefrontal cortex, leading to increased thinking, narrowly focused attention. Indica varieties, on the other hand, suppress prefrontal cortex activity, resulting in reduced thinking and planning, relaxation and sleep promotion. Regardless of the strain or THC/CBD ratio, cannabis use generally suppresses neural circuits in the basal ganglia, involved in action planning and go-no-go circuitry. Cerebellum, responsible for balance, motor planning, and motor sequencing. Common effects of smoking marijuana, regardless of the strain, include reduced physical mobility, reddening of the eyes, dryness of the mouth. The latter two effects are caused by a reduction in saliva and tear secretion due to the presence of CB1 and CB2 receptors in the mouth and eyes. Some strains of cannabis can also increase appetite, commonly known as the munchies. This is due to the high density of CB1 receptors in the hypothalamus, particularly in areas like the arcuate nucleus. When THC and CBD bind to these receptors, they activate neurons that stimulate appetite through two mechanisms. 1. Cognitive mechanism, creating a preoccupation with food, anticipation of taste, and the experience of taste. 2. Physiological mechanism, stimulating appetite through the activation of specific neural pathways. These mechanisms demonstrate the complex effects of THC and CBD on the body. Additionally, the widespread presence of CB1 receptors in the nervous system, including the spinal cord, contributes to the potential pain-relieving properties of cannabis. While some people may perceive significant pain relief from cannabis, research suggests that this effect is partially due to a shift in perception, where the individual focuses on other things. However, cannabis, particularly THC, does exhibit some genuine antinociceptive or anti-pain effects. These effects are primarily mediated by CB1 receptors in spinal cord neurons. Regardless of the strain, consumption method, or form, cannabis use triggers a wide range of effects. This diversity of effects can be attributed to the retrograde signaling mechanism, which can either activate or suppress neuronal activity in various parts of the brain and body.